My name is Blake Collins, and today I'll be discussing the group of neurodegenerative disorders collectively known as motor neuron disease. To start today's presentation, here's a quick overview of what we'll be focusing on today. Firstly, the pathophysiology, which will be followed by the typical signs and symptoms and their effect on movement control, treatment and interventions, which are currently in use or being investigated, and the benefits of exercise and guidelines for exercise prescription. Before we get into the pathology that affects motor neurons, we'll first overview the system in question. With help of the diagram to our right, we can see that upper motor neuron cell bodies are situated in the motor cortex and project axons via the corticospinal tract to the spinal cord. There they synapse in the anterior horn with lower motor neurons, which project axons via peripheral nerves that then contact muscle fibers at the neuromuscular junction. Motor neurons are the sole means by which skeletal muscle contractions can be triggered and thereby control the movements on which life depends, which include swallowing and breathing. The term motor neuron disease was first proposed to incorporate a variety of related neurodegenerative conditions into a single spectrum of disorders. They all have considerable disability and poor prognosis and include amyotrophic lateral sclerosis, primary lateral sclerosis, progressive muscular atrophy, and progressive bulbar atrophy and palsy. Amyotrophic lateral sclerosis is the most common motor neuron disease. Its epidemiology is between 2 to 5 per every 100,000, and it's a progressive neurodegenerative disease that affects both the upper and lower motor neurons in the motor cortex, brainstem, and anterior horn of the spinal cord. Amyotropic refers to muscle atrophy, weakness, and fasciculation, which is indicated in the diseases of any lower motor neuron. And lateral sclerosis refers to the hardening of the anterior and lateral corticospinal tract as motor neurons degenerate and are replaced by gliolysis. Risk factors include genetic, with approximately 5 to 10% of ALS cases inherited, age, and sex. The multifactorial nature of the disease leads to a variable clinical course. The mean onset of disease is 58 years, with near linear neurodegeneration resulting in death. Death usually occurs within 1 to 5 years and is usually because of respiratory failure. While first reported in 1869, the exact cause is still relatively unknown. Exogenous environmental factors that have been implicated include toxins, infections, exposure to heavy metals or chemicals, and mechanical and electrical trauma. Also, controversially, rigorous athletic activity. However, the focus of our presentation today will be on the genetic SOD1, exotoxicity, mitochondrial dysfunction, and oxidative stress that all could potentially result in apoptosis, which is the programmed cell death of neuronal cells. Firstly, oxidative stress. It's a pathological process representing an imbalance of normal metabolism. Its detrimental effect is caused by the reactive oxygen species, which cause radical production and or failure of protective deoxytizing or repair mechanisms. The oxidative stress is a major contributor to ALS pathology and it affects presynaptic transmitter release as well as contributing to the retrograde degeneration of neuromuscular junction. Mitochondrial dysfunction. In addition to its energy production role via oxidative phosphorylation, mitochondria have roles in calcium, homeostasis, free radical production, and apoptotic roles. These features are all shown to decline with age, which, if it is involved in the production of ALS, would explain the high incidence of ALS with age. The combined effect of increased ROS production and mitochondrial dysfunction ultimately could lead to cell death in tissues and organs that are most dependent on oxidative phosphorylation for survival, as neurons depend exclusively on aerobic metabolism, making them more vulnerable to mitochondrial defects and toxic effects of mitochondrial free radical generation. Glutamate exotoxicity. Glutamate is a crucial excitatory neurotransmitter, and in higher than physiological normal concentrations, it can overstimulate receptors, causing abnormal calcium ion influx into neuronal cells and ultimately neuronal cell death. One of the biggest supporters of glutamate exotoxicity's role in ALS is the success of clinical trials of riluzole, which will be discussed later in the interventions part of the presentation. SOD1. Mutations in the CUZ and superoxide dismutase 1 gene is associated with 20% of familiar ALS. It plays a role in the cellular defense against oxidative stress via catalyzing the conversion of superoxide anions into hydrogen peroxide, which is further metabolized. It's related not to the loss of function, but to the gain of one or more toxic properties. There's over 100 mutations of SOD1 genes that have been identified. However, the type of mutation does not correlate with the severity of the disease, and some mutated forms have actually maintained normal function within the body. There's an experiment on mice to manipulate the SOD1 mutation and look at the effect that had on ALS. They had two sets of mice. One had the SOD1 gene knocked out altogether, and the other was a transgenic mice with an overexpressed mutated SOD1 protein. In the mice group with the SOD1 gene knocked out that didn't develop ALS, however, in the transgenic mice with an overexpressed mutated SOD1 protein, there was a development of adult-onset progressive motor neuropathy, which had a phenotype similar to ALS in humans. Testosterone. The role of testosterone in the central nervous system is still unclear, however it is believed to be associated with regulating motor neuronal survival and axonal regeneration after injury. It regulates dendritic length on adult motor neurons and exerts an anti-apoptotic effect in cultured neurons. A study found that free testosterone was significantly lower in both men and women who had ALS. Signs and symptoms. The hallmark of ALS is progressive, selective dysfunction and death of motor neuron pathways. The signs include asymmetrical weakness and wasting in the limbs associated with clinical evidence of corticospinal tract damage, which includes increased tone, brisk reflexes and extensor planters, and the disease usually begins either in one limb, foot, for example foot drop, or wasting of the intrinsic hand muscles, or with a combination of bulbar and corticospinal symptoms, dysphagia, dysarthria, tongue wasting, and brisk jaw jerk. Movement control. Degeneration of upper motor neurons leads to increased muscle tone and extensor plantar response known as spasticity. Spasticity is a neuromuscular overactivity caused by upper motor neuron degeneration. Overactive neural input to the muscle causes excessive contraction. In motor neuron disease, the primary mechanism behind this increased tone is hyperplexia. Absence of corticospinal inhibition onto lower motor neuron causes the subsequent development of lower motor neuron excessive excitability, which in turn creates an excessive response to muscle spindle input. The loss of inhibition results in involuntary muscle contraction that can cause stiffness, which then reduces range of motion, that interferes with speech and locomotion. Common clinical manifestations include hypertonicity, clonus, which is involuntary contraction, and fixed or frozen joints and spasms, which are related to pain. The lower motor neuron effects include muscle atrophy, reduced or lost tendon reflexes, which is commonly referred to flail arm. It is the loss of lower motor neuron function that leads to the paresis, paralysis, and the eventual death. Treatment and interventions. It would be unethical to apply some of these treatment interventions to humans, so mice are generally used. The reason behind this is that mice harbour a similar transgene expression human cytosolic superoxide dismutase with an ALS-associated mutation, which results in the development of motor neuron disease that closely mimics the phenotype of both familial and sporadic ALS. So the thought behind these interventions is that if it works on a similar phenotype, then it should work in humans. The first intervention we've got here is the use of insulin-like growth factor. Insulin-like growth factor 1 has been suggested as a possible intervention in therapy of ALS because it stimulates survival and axonal growth in motor neurons and protects skeletal muscle fibers from atrophy. Using the mice models, injections of IGF-1 was used in both the mild and severe disease progression in familial-based ALS. As we can see in the graphs here, a substantial delay in symptoms onset combined with improvements in body function, motor performance, and animal survival was achieved after the IGF-1 treatment in the slow progressing disease. In contrast, the fast progressing disease model, there was no significant therapeutic relevant improvements in the tested parameters. The second intervention we'll be looking at is the activation of transcription factor 3, which is a basic leucine zipper transcription factor. In the body, it's induced under a variety of stressful conditions. ATF3 transcription targets vary in different cell types and conditions, resulting in diverse influences on cellular phenomena. It can either have a role in cell survival, proliferation, and death. In neurons, induction of ATF3 expression prevents cell death and promotes neurite formation and elongation, leading to enhanced nerve regeneration through transcriptional induction of survival and growth associated genes. The current study that was used for this in 2014 was the injection of ATF3 in ALS mice. As we can see in the graph here, in the ALS mice, they showed a slowing of loss of motor neurons. delays terminal axonal atrophy, and augmented axonal sprouting by driving the motor neurons into a pro-survival and regenerative state. In the ALS mouse model, forced ATF3 expression modifies the cell transcriptome to support compensatory axonal sprouting and neuronal survival. After 90 days, the ATF3 injected mice showed no loss of neuromuscular junction innervation, despite some motor neuron loss, suggesting that neuromuscular junctions that had previously lost innervation were rapidly reinnervated by terminal and collateral sprouts extending from neighbouring intact motor neurons that are in the pro-regenerative state. The third intervention is the actual only treatment that's currently used in human trials, which is known as riluzole. Its role is to inhibit glutamate release from the presynaptic terminals of neurons. The dose of 100 mg per day of riluzole has been shown to increase lifespan by an average of 2 to 3 months. While there's been success in the mice models of both the insulin-like growth factor and ATF3 models, as well as small improvements in life expectancy with riluzole trials in humans, the current best intervention remains to be exercise benefits. The role of physical activity in ALS, however, is very controversial. Physical activity is recommended in a variety of neuromuscular disorders and has been successful in the majority of them. However, the implication of intense exercise before the onset of disease, which is linked to reactive oxygen species production and glutamate exotoxicity, which, as we know from our previous slides, have a indicated role in the production of ALS. Therefore, there's a hesitation to promote exercise. The thought behind physical activity being beneficial to neurodegenerative disorders includes the brain-derived neurotrophic factor stimulation, which is related to neural health and neural excitability. Brain-derived neurotrophic factor plays a fundamental role in neuronal health, with roles in synaptic plasticity, neurogenesis, and adaptive behavior, and protection against injury and disease. Neurons that are relatively inactive and/or receive insufficient target-derived neurotropic support undergo programmed cell death. Conversely, neurons that are electrically active and interact with other target neurons are more likely to survive. With all this in mind, and the absence of a cure, the aim of the exercise program is to improve quality of life. In the study done in 2001 by Drury and his colleagues, they implemented a moderate range of motion exercise program and it was shown to have increased muscular endurance. These increases were thought to be due to increases in oxidative metabolic potential of the muscle. It was also shown to increase strength and delay the decreases in functional rating scales. Exercise guidelines. As we discussed before, the high intensity resistance training dramatically increases free radical production, and any kind of strenuous exercise that induces oxygen consumption will increase free radical generation during exercise. Therefore, one of the guidelines should be to use light to moderate intense exercise and avoid vigorous intensities at all costs. The most successful exercise programs so far have been range of motion, so that would be another guideline that would be to use range of motion exercises as opposed to aerobic and/or resistance type training. ALS patients have also been shown to have decreased grip strength, so any exercises that involve gripping or manipulating of bars are going to be difficult. And there's also a high dropout rate generally associated with the pain that comes from exercising, so that's another guideline to be aware of. What are our take-home messages from today? The take-home messages from today is that motor neuron disease is an umbrella term. It's given to incorporate a variety of related neurodegenerative conditions into a single spectrum of disorders. Of these disorders, ALS is the most common. However, we're no closer to knowing the cause and what consequent treatments can be. However, there is growing support behind the mitochondrial dysfunction leading to apoptosis, as well as the clinical trials of IGF-1 and ATF3 and riluzole are promising. Exercise, while prescribed in a variety of other pathogens as a viable management strategy for neurodegenerative diseases, remains a controversial topic with some improvements found. Thanks for your time. My reference list can be found on the attachment. Thank you.